Now that I've given you the big picture of central dogma, we will now focus specifically to the first step, which is called as transcription. During transcription, a segment of the DNA which serves as a template for the production of an RNA molecule will begin the process. And this segment will first transcribe this RNA molecule, which will then leave the nucleus. So we will just go ahead and look at the first couple of steps. The first step in the polypeptide production occurs in mainly, must remember that this is taking place in the nucleus. So that's where the whole process will begin with. There are certain key players or certain components that help this process start and complete. The first thing you must have is a DNA coding strand. The DNA coding strand is your template strand. If you don't have that template strand, the RNA polymerase, which is the enzyme, the purpose of this enzyme is to first come and join, read the nucleotide sequence on the DNA code and start putting the nucleotide to the 3' end. Again, it will join the nucleotide from the 5' to the 3' directions. You must have the nucleotides, which are, you must have A, G, C, and U. So without that, it's not going to work. So let's just put down, this is the DNA strand, and RNA would be, let's say this is the enzyme. The RNA polymerase is the enzyme. Must have ATP molecules because certain bonds are formed and certain bonds are broken, and which requires the energy. And most importantly, it must have a region on the DNA which is called as the promoter region. A promoter is a region where the gene will start to be transcribed. So RNA polymerase is not just going to go anywhere in the DNA and start making copies of it. It recognizes certain regions on the DNA, and that region is the starting point, and that's where the promoter is present. And we're going to see an animation on this one too. Hopefully the animation will help you understand what a promoter does. So now once a promoter is recognized within the DNA strand, the RNA polymerase starts getting busy and starts joining the nucleotides. Now once the RNA polymerase starts making the nucleotide within the promoter region, it's actually going to have a code which will say start the process, and that's called as the start code. So there is a start code which helps the nucleotides to recognize the promoter. And similarly, there is also a stop code, because if there was no stop code, the polymerase would just keep on making a long strand of messenger RNA. And that is called as the stop codon. So if you recall, if you go back to the chart that I showed you where we had all the 64 combinations, you will find a start and a stop code over there. Now, at the end of this process, there is this purple region which is coming out is the messenger RNA. And this messenger RNA is now considered to be an immature messenger RNA. The word we use immature is because this messenger RNA can be hydrolyzed by certain enzymes as soon as it leaves the nucleus. So to protect this, there is a process called as mRNA processing, and we will talk about this later in the coming couple of slides. But this would be the main process of transcription. So let's go on to dividing the main steps of transcription into three processes. First process would be called as the initiation. That is the binding at the promoter. This is the start of the process. This is where the promoter region will be recognized. Stop codes are read, and RNA polymerase starts elongating the mRNA. Elongation is the main process where the mRNA is now growing in length, and eventually you will reach the stop code, which will then stop the sequences. Now, let's talk about how does the mRNA becomes mature. mRNA becomes mature is because there are a couple of things that goes on within maturation of the mRNA. So let's look at some of the modifications that can happen. Also keep in mind that these modifications are only taking place in the eukaryotes. The prokaryotes has a much simpler process of maturation. The first thing I want to talk about is actually the introns being removed. Now, when the transcription began, and hey, where is the DNA, and it's making it, transcribing it into the mRNA, it starts with the start sequence and ends at the stop sequence. Now within that, it has regions that actually have no sense, and these are called as the introns. These are regions of the DNA that has been copied, but it does not really give any particular protein to be formed. Mainly like a nonsense regions you can think as. The introns are the nonsense regions, and these have to be removed. So there are enzymes that will come and chop these introns away, and once their introns are removed, it starts joining the exon, which are the coding sequence. So now if the length is this long over here for the immature mRNA, the mature RNA is going to be shorter because the introns have been removed. So that's number one. Secondly, what happens is a region which is the 5' end is then a cap is added on this region, and the purpose of the cap is to stop the enzyme from self-destructing this mRNA when it leaves the nucleus and enters the cytoplasm. And thirdly, on the other end, which is the 3' end, a poly A tail, which means that there is numerous adenine nucleotides that are added at the end, and this is the poly A tail. The purpose of the cap and the poly A tail is mainly to inhibit degradation of the mRNA by the hydrolytic enzymes that are present within the cytoplasm. I usually give students an analogy for like a shoelace. Think of the shoelace and think of the two little caps that we have on the shoelaces. If you don't have those caps, and the lace starts fraying. So think of it the same way. This process of the mRNA to be more mature and to be self-sustaining and not to be destroyed by the enzyme are all parts of the post-transcriptional modifications. Now, once the mRNA has removed its introns, it's got its 5' cap, and it has a poly A tail, it's ready to leave the nucleus through the nuclear pores and into the cytoplasm. And now, what happens in the cytoplasm? That's as part 2 of this process, which is translation, and which we will learn in the next in the series 14. Here I just want to go over quickly something about the introns before I leave you. The introns are pretty much the nonsense, or they used to be called as a junk DNA, but now there is a very interesting concept as to what this junk DNA does. This junk DNA actually helps to identify genome from one individual to the others. So how do we differentiate between individuals is basically based on certain sequences that are present within the junk DNA. In humans, 95% of the DNA is mainly made up of these introns. So that's really the junk. You only have less than 5% of the DNA, which is actually the sensible or the coding DNA. So in particular, the eukaryotes, the primary mRNA processing is done before the mature RNA leaves. So we will stop here, and once we've understood transcription, we'll later on study translation in the series 14 of this semester.